The purpose of the Posner Cueing task is to assess attentional shifts in space while holding fixation on a given locus. Attentional shifts occur when attention is directed on a certain location or point. Subsequently, there is a processing priority given to any stimulus within the location which attention is focused. The Posner Cueing task assesses whether providing participants with a target that is in the same location as the cue providing to the prior can impact reaction time and the amount of correct responses given. Valid trials are trials where the target is displayed in the same location as the cue that was presented to them. Neutral trials are trials where there is no cue and thus there is no attentional orientation to either provided location. Invalid trials are trials where the target is displayed in the opposite location as the cue they have previously been shown. It is hypothesized that valid trials will have the fastest average reaction time, while invalid trials will have the slowest reaction time.
So these were the methods used to test our hypothesis. This experiment had a within subjects repeated measures design where all participants were exposed to all conditions or trial types. In all three of these trial types, subjects were asked to remain fixation at a given location, which is represented by the small cross in the middle of the first box in the paradigm figure. They were then cued to a locus to the left or right of the fixation point, represented by a small green X in the second box. The neutral trials, however, had no cue. Next, participants completed a target detection task where a target appears in one of the two boxes to the left or the right as seen in the last box. The participants were instructed to respond with whether the target appeared on the left or right as fast as possible by clicking one of two keys on a keyboard. In this case, the three conditions or trial types were valid, neutral, or invalid, and each participant completed many trials of each of the three in a random order. The valid condition was where the cue location and the target locations matched, so they are both either to the left or to the right. The invalid condition was where the cue location and the target locations did not match, so they are on opposite sides of the fixation point in the middle. And as I said before, the neutral condition had no cue.
So the software used for this experiment measured the amount of time it took each participant to respond with the target location from the time the target was presented to the time the participant responded by pressing the button. The keyboard acts as a response mechanism for the participant. This was also recorded along with each trial type, valid, invalid, neutral, and the participant ID. This task measures a participant's manual movement as well as eye movement in response to a target stimuli in order to demonstrate the effects of covert orienting attention in response to the different cue conditions. This measure is used as a basis to differentiate reaction times across the three types of trials. There is a set interval between each type of trial, usually ranging from 2,500 to 5,000 milliseconds, that is predetermined by the experimenter.
Our results. We took the raw data from the Posner Cueing task collected during class periods and analyzed it. The accuracy of the responses recorded was divided up into accurate, inaccurate, and timed out. An accurate response was when a participant would indicate the correct placement of a target stimulus. Accuracy was only based on the position of the target stimulus, regardless of where the cue had appeared before it. A timed out response was where a participant did not answer within 1.5 seconds, or 1,500 milliseconds. We also filtered out any responses that were recorded less than 250 milliseconds after the target appeared, because visual information takes at least 250 milliseconds to make it through the visual system. Therefore, any responses recorded that were faster than that were likely reflexive button pushes or accidents, and not actual responses to target stimuli. Of the data left over, we were left with only those responses where participants strive for accuracy while still being as quick as possible. By then dividing up the reaction times by cue validity, we were able to see the average response time for each of the three validities, as shown in the bars of the graph. The error bars are from the standard error of the means. There was a significant difference between average invalid and valid cue response times, as well as a as well as a significant difference between the average invalid and neutral response times.
Now, when it came to analyzing the data, our results were pretty clear. As we investigated locational cues and their effects on finding changes in an object that has been previously seen, it was very evident that our participants still had their attention drawn and their ability to identify distinctional environmental changes also quickened. These results were all dependent on the cue's validity. Our participants' quickest response times came from valid cues, and our participants' slowest response times came from invalid cues. A study such as this one has the potential to extend beyond just simple processes of complex finding, and could even go on to improve the well-being of those in society overall. One specific group that could benefit from this study is children. Specifically, this form of study could tell you how to draw their attention from one location to another, and it could even have implication on the cues necessary for creating more successful academic videos and future children's educational software. Another way this study could help is through neuronal activity. Locational cues interact with visual pathways, which is connected to brain processing. So this kind of study could inform us on which visual stimuli have the greatest effects on neuronal firings.
The motivation behind our follow-up experiment was to examine whether the results found in the original study would also apply to a more complex level of attentional processing. To explain this using the paradigm shown here, subjects would see a picture for 3 seconds, that would then be followed by a fixation, and then a cue would occur on either the right or the left side of the fixation. After that, participants would see the picture again, and they would have some kind of change on it, whether that be an addition like the one shown here, or an elimination where we take something significant away from the picture. And then they would need to press whichever keys we chose to correspond with the left or the right side to indicate which side they see the change on. And then as the experiment progresses, we would make the change more and more subtle to see how that would affect response time, and at what point would response time maybe be affected more by the fact that the change was so subtle it was difficult to see or to recognize right away, rather than whether their attention was cued in a valid or invalid trial. Based on the results of the original Posner Cueing task, what we would expect to happen in this experiment is that participants would react more quickly to a valid trial than an invalid trial, and this trend would continue over time, even as the changes became more subtle. However, as the changes became more subtle, what we would expect to happen is that the distinction between response time to valid and invalid trials would become less obvious, just because it would be more difficult to spot the change, which would result in a longer response time regardless of valid or invalid trials. But overall, they would still continue to respond more quickly to a valid trial than an invalid trial. With that said, that is the end of our presentation. Thank you all for listening.